then I get something which converts rate 1. And so that tells me that in this sort of scaling limit, in the large population limit, I get pairs of lineages which coalesce at rate 1. There's nothing special about any particular pair of lineages. And so if I track the ancestry of a larger sample, then all pairs of lineages coalesce at rate 1. So the total rate of coalescence when I have k lineages remaining is k choose 2. And what I end up drawing for myself is this sort of random tree where these holding times are exponentially distributed with rate k choose 2 while there are k lineages. This retrospective model ends up being particularly useful for my purposes in the sense that I want to think about sampling from population and doing inference based on those random samples. And this thing encodes exactly the history of a sample of lineages rather than looking at the whole population. So in that sense, morally, it's a good model to look at, but it's also a complicated one because these ancestries are unobserved. In virtually all cases of biological relevance, what you usually get is you get a sample which you can think of as coming from the leaves of that coalescent tree on the previous slide. And the pattern of SNPs along these lineages, I'm thinking here of an infinite sites model of mutation for the people who know what that is, but that's not a particularly important detail. None of what I'm saying is crucial for the infinite sites. In any case, the pattern of diversity of SNPs and of mutations you see is somewhat informative of the tree, but doesn't give you full information from the tree. On the other hand, if you could see the ancestral tree of these individuals annotated with when the mutations happened, then your job would be easy. Doing inference in the genetics context would be largely a solved problem. You could get away with statistical techniques which are the better part of a century old now, and you would have to do a little bit of coding to store things cleverly, but your job would be a pleasant one, let's say. But of course, you can't get the tree. I mean, in some limited cases, maybe now we're beginning to have enough data that you have some fairly good information about what the tree looks like. But in the majority of applications, especially in an organism like humans, you're not going to be allowed to go and exhume quite that many bodies. You probably wouldn't want to, even if you were allowed to. So the approach you take is to think of the ancestry as missing data. And you're left with the task of evaluating a likelihood function. So this I'm defining rather implicit notation. It says probability of the data. I don't have access to this thing. I only have access to it if I know the tree. So what do? I pretend I know the tree. I look at the probability of the data given a genealogy and then average over all possible genealogies. And the Kingman coalescent gives me the model that I use in order to do this averaging. So this P of DG here, which is averaging over the genealogies, is the distribution of the Kingman coalescent. There isn't a hope of evaluating this integral. It's over the space of binary trees with a fixed number of leaves. It's far too large to calculate analytically or with most numerical methods. So in practice, if you want to do something which is sort of approximating this in a rigorous sense, for instance, saying that if you give it an arbitrarily large number of computational power, you get what's the correct value of the integral out at the end, then essentially you're forced to use Monte Carlo methods. There isn't another feasible way to tackle this integral. Arguably, Monte Carlo struggles to be feasible as well, but it's the closest thing we have. And what you end up doing is you end up averaging over an ensemble of trees drawn from this distribution. And you treat this ensemble average as the true value of the integral up to some error. In order to be able to do this, you have to be able to store and manipulate the ancestral structures. And for the purposes of this sort of simple model with one tree, that's not too difficult, but I'll show you how to do it in this case, and then we'll come on to talking about recombination. So once you have trees, if you have a tree, you still need to be able to generate those, the sort of replicates in that ensemble sensibly. And the standard, pretty much the standard workforce for how to do that is called Markov chain Monte Carlo, in this case, more specifically the Metropolis-Hastings algorithm. I'll introduce that briefly before I go into the details of the data structures. So for the purposes of this slide, in principle, you can forget about trees. This works in fairly arbitrary state spaces with fairly arbitrary statistical problems. And it's a way of generating good candidate trees in order to, or good candidates in order to do this approximation. If you just sampled Kingman coalescent trees naively, you would end up, for various reasons, getting a terrible approximation out of this. So instead, what you want is you want to find one good tree and explore a region around it. The Metropolis-Hastings algorithm does that. It starts from a fairly arbitrary initial condition and iterates the following dynamics. I propose a small perturbation of my current state of the algorithm, and then I either accept that perturbation and my chain moves into that position, or I reject it and stay where I was. So this is the probability with which I want to accept my perturbation. What is this? Well, this is my target distribution. This is the distribution I want to sample from. So in the case of the previous slide, pi would be the distribution of the Kingman coalescent given the configuration of mutations. And the best way to see what this accept-reject probability is doing is to assume that the perturbation kernel Q is symmetric so that it cancels out. Then what I see is if I've proposed a state which increases the distribution from which I'm trying to sample, increases my likelihood, if you like, then I always accept that proposal. On the other hand, if I propose going downhill, moving to a tree which is slightly less likely under the distribution I want to sample, I still want to accept sometimes in order to be able to explore the state space, but it's with a probability strictly less than 1. So this is the right way to balance exploration and exploitation in such a way that your samples, if you were to be able to run this chain forever, the empirical distribution coming out of a time series from this chain would exactly coincide with distribution pi. It's a one-line calculation to do to check that that's the case, provided that your perturbation kernel Q is not completely pathological. It has to be it can be more or less whatever in theory, as long as it's not completely pathological. Of course, its design crucially affects the actual efficiency of the algorithm, whether this actually runs well in practice. Okay, so I want to be able to store ancestral trees in such a way that I can, with reasonable computational effort, do these kind of Metropolis-Hastings perturbation steps on my space of trees. That's the task for the time being, and this is one slide which tells you how to do that. This is the kind of cartoon of trees which is easy to think about, but not immediately obvious how to code onto a computer. And this matrix turns it into something which a computer finds easy to handle. So this is just a matrix of numbers. And what's going on here, the index at the top, you can think of as the indices of my nodes on the tree. So indices 1 through 4 are leaves, 5, 6, and 7 are ancestors. Pi tells me what is the ancestor of the corresponding leaf. So the ancestor of 1 is 5, the ancestor of 2 is 5. That's this coalescence event over here. 1 and 2 don't have children, so this empty set is just a null symbol, meaning there's nothing there because they're leaves. And the branches they subtend start life at time 0. On the other hand, individuals 5 and 6, they have a common ancestor in 7. That's this event over here. They have children 1 and 2 for 5, 3 and 4 for 6. You can see that that just helps you propagate downward along the tree. And the nodes live at times T5 and T6. So T5 would be this height, T6 would be that height. And this table tells you everything there is to know about this tree. They are in a one-to-one mapping, except this is something that is easy to code. And that's the whole point of this. These two lines on the bottom are, this is the distribution of the Kingman coalescent. These T sub i's in brackets are the order statistics of the times of the nodes. And the point isn't the exact functional form, it's simply that this distribution can be evaluated from the information in this table. Right, and the same is true of the second line, which is the probability of mutations given the tree. It's again something which you can evaluate given the information in this table, plus a little bit of information about the mutations, the pattern of mutations that you've seen in your data set. So I'm not going to unpick these formulas, just to emphasize the point that they can be evaluated from this table, and these are the quantities we need to evaluate in order to run our chain, so life is good. How do we actually do something like a Metropolis move on this state? Well, a common approach would be something like a Subtree, prune, and regraft move, in which we take one branch randomly selected, break it off from the rest of the tree, and reattach it somewhere else. In this diagram, I'm breaking 2 away, and I'm reattaching it into the lineage above 6 over here. What happens to my actual table? Well, the times are pretty simple. So, well, actually, let me return to the times and go here first. What happens is node 5 disappears from above node 1. It isn't needed there anymore because there's no merger event there. Instead, 1 connects directly to 7. And you can see that the parent of 1 moves from 5 to 7. The parent of 6 moves from 7 to 5 because there's now a new node being created above node 6. The label 5 happens to be free. It doesn't matter that it's numerically higher than 6. And so I'm using that in order to encode the new node above node 6. And that's the only change that's needed over here. In principle, the ancestor of 5 might change as well. In this example, it doesn't because node 7 happens to be the node that's above it in both before and after the event. And the timing of node 5 changes as well. But I can perform this operation, changing the tree, by changing an order 1 amount of information in my data structure. So the amount of change I have to make here does not depend on what my sample size is, for instance. And this I could use to run my Metropolis-Hastings chain. There's a further advantage in this tree-based approach in that it makes storing ancestries very efficient, storing data very efficient, in fact. If you imagine the storage cost of storing a binary incidence matrix, well, you have n samples and m sites. The size of the matrix is n times m. And under sort of usual human-like scenarios, the number of sites will be something like the number of SNPs, rather, will be something like the number of sites you've looked at times the log of n, just because that's the total amount of branch length in the tree. So overall, you get something n times L log n. On the other hand, if I store my tree, well, it has order n branches. It's a binary tree with n leaves that has about 2n branches. So it's linear in the sample size. Once I have a tree, I can put my mutations, just decorate them on top of the tree. And I need to store each mutation once somewhere along the edges of the tree and say that it's present in all of those samples which subtend it. That's an order m operation, or L log n. I don't know why that n's capital, it shouldn't be. And if I add these two up, I get n plus m, or n plus L log n, which is considerably smaller than what's over here with the product. And so even if nothing else has happened, the fact that I've actually constructed a tree from my data set and decorated the tree with the mutations has made my data set much, much easier to store. Its storage cost scales much better than it would as a binary incidence matrix, for instance. And that alone can give you a considerable performance boost for various sorts of analysis algorithms. All right, so I promised to talk about recombinant ancestries. I've used up half of my time without mentioning recombination, so I had better rectify that. And now all I'm going to do is complicate my model a little bit, put in recombination, and then think about, well, what do I have to do to my ancestral structure in order to incorporate recombination in the picture? I'm conscious of the fact that there are people who know more about biology than I do in the room, so you're going to have to forgive me. I'm only going to talk about meiotic crossover recombination and probably a simplified case of that. Similar ideas can be extended for other forms of recombination, but I won't go into that for the purpose of the talk. So for me, when I talk about recombination in a coalescent model, what I mean is I want to go back all the way to that simplest Wright-Fisher model that I started with, but I want to now think about diploid individuals and think about a single haplotype from such an individual and think of what happens to that backward in time. And I want to have some per generation recombination probability R, which scales inversely with the population size. With probability 1 minus R on n, I get a simple picture in which nothing happens. I copy my haplotype from one randomly chosen parent wholesale. With the complementary probability, I have a break somewhere along my haplotype, and I copy one half from one parent, one half from, well, one half from the other copy on that parent, if you like. I could also take my number of breakpoints to be Poisson distributed with the same parameter. That would end up giving me exactly the same scaling limit. So the fact that I'm only allowing one recombination in my cartoon isn't in practice a limitation relative to this scenario. Excuse me. So what happens to my ancestral structure as I have one of these recombinations in the picture? Well, I start with the same number of, with a number of sampled lineages again, and backward in time, I now see two kinds of events. I see merger events, for instance, lineages 4 and 1 merge into a common ancestor at 6. That's exactly as it was in the Kingman coalescent. But I have a different kind of event, which corresponds to recombination. So lineage 2 has a branching event into two halves, lineages 4 and 5. I've represented my tables in slightly a different way, which happens to be convenient for recombination, but these contain exactly the same information as that previous table in my picture without recombination. So how do I read this? Well, if I look at edge 2, edges 2 and 3, which are the two edges where 2 is a child node. So 2 is the leafmost endpoint of these two edges. You can see that the identity of the parent differs, and it differs based upon where along the segment I'm looking at. So I'm thinking of my whole haplotype as just being the unit interval 0 to 1. So any edge on which the full segment 0, 1 is covered contains no recombination. But over here I have a breakpoint x. Everything to the left of x goes to 4. Everything to the right of edge x goes to 5. And that material gets tracked further back along those edges. So edge 4 goes to parent 6 over here. And again, the material that I've transmitted is only 0 to x, because that's all the ancestral material I care about. As in the previous slide, of course, these parents still contain a full copy of the haplotype, but this dashed material is non-ancestral. I don't care about where it goes, so I don't model recombinations in that material anymore. And this representation ends up being a very compact way to specify recombinant genealogies. But in fact, you can do a bit better still. You can do a bit better by thinking about, what do we actually know? What's identifiable from mutation data? So suppose I have an infinite sites mutation model, and I have a very high mutation rate, so I see all merger events. Even so, I still would not be able to date this recombination event over here, because whether a SNP happens on the lineage connecting 4 to 6, or in the same material connecting 2 to 4, I won't be able to tell the difference from just looking at the leaves. And so what I can actually see from data is this picture. This is the same realization of a recombinant ancestry, but stripped down to its most basic ingredients in terms of what is actually observable from data. I no longer see the recombination event. I only see that eventually lineage 2 merges with parent 4 in one half and parent 5 with another, but I don't know exactly where the recombination happened. Everything else is exactly the same. The interpretation of this table is exactly the same. The table is somewhat smaller because there's fewer nodes involved. I don't need to put in those extra nodes in order to encode the recombination event. And as a consequence, I get a somewhat smaller data structure. There ends up being a downside, though, which happens if you look at the functions, the likelihood functions you need to be able to evaluate in order to do Metropolis-Hastings sampling for these so-called ancestral recombination graphs. So these are the analogs of the same distributions that I had on the Kingman coalescent slide. And again, I don't want to unpick the formulas in a great amount of detail, except to say that this bottom one, the probability of data given the genealogy, that I can evaluate from that reduced, simplified genealogy. But this top line, I can't. And that's the ancestral recombination graph prior distribution, if you like. And I keep dropping my microphone, excuse me a moment. There we go. What this one needs is, at a minimum, in each time interval, in each holding time of this ancestral recombination graph process, you need to know how many lineages are there and how many links in those lineages are what you might call active, in the sense that if a recombination were to happen there, it would separate at least some ancestral material into two chunks. And in this simplified picture, I don't have that, because I know at some point in the time from the leaves to the time to node 4, there are 3 lineages, but at some point that turns into 4, because there is a recombination event in there somewhere, and I don't know where it happens. And therefore, I do not have the number of extant lineages and the number of extant links at all times for my realization. I can't evaluate this distribution. And so if I want to do Metropolis-Hastings sampling, at least without doing something particularly clever in order to try and overcome this problem, then I can't use that simplified genealogy. I have to use the bigger unsimplified genealogy, not because I need to evaluate my mutation likelihood that I can do from either one, but because of the ancestral recombination graph model itself. What's the consequence of that? Well, let's look at the kind of storage cost of these tree sequence-based models in the context of recombination. So again, if I store my data as a binary incidence matrix, well, that doesn't care about recombination going on. The binary incidence matrix has the same cost as it did in the Kingman coalescent case. How do I store a tree sequence? Well, I have to store an initial tree for, say, the leftmost point of my genealogy, of my chromosome, I should say. And then I have to store transitions through trees. So a recombination will essentially enact one of these subtree prune regraft moves and change the tree in that way. And so I already highlighted earlier that a subtree prune regraft can be implemented in order 1 time. A single one of those will require order 1 computation or order 1 storage. How many recombinations do I actually get? Well, if I look at my simplified case, then it's known, this is, I think Jotunhein's book is where these sorts of results originate from, that the number of recombinations which change the tree, it is an order L log n quantity, similar to the mutation, essentially the number of recombination events that you get along a single tree. SNPs given a tree, well, these you can just decorate on the ancestral graph again. There, you just have to place each SNP once somewhere along the graph. And so overall, you get initial tree plus number of recombinations which change the tree, plus this SNPs given the tree, and this is your graph-based storage. And again, the kind of sample size and the chromosome length have effectively decoupled up to log terms. On the other hand, for the unsimplified graphs, it's worse. There's this square on top of the chromosome length term, and that's the thing that really kills you, which if you plug into here 10 million for 10 megabases or even more than that, and then you raise that to a second power, that's just not the sort of number that computers are capable of handling anymore, no matter what you do in terms of software engineering. You're not going to get the constant small enough. And so I think this square here is what puts a fundamental limitation onto what sort of theoretically sound MCMC-like methods can actually do in the context of recombination. So having kind of found that there is this fundamental limit, we still wanted to see how far you could actually go, because one thing that these MCMC methods are very good at doing is they give you very nice quantification of uncertainty. And given that these kind of ancestral recombination graphs are very poorly identified by datasets, you certainly can't pin down a single graph structure from a recombinant dataset, then uncertainty quantification becomes essential. This slide briefly summarizes the sort of Metropolis-Hastings moves that we tried. So there's a collection of different kinds of perturbations of the underlying graph that we tried. These are all classical things present in the literature. There hasn't been anything clever here. All we've essentially, what we wanted to focus on is what is the effect of using one of these optimized data structures from the MS Prime TS-Kit world on Metropolis-Hastings based sampling. So subtree prune regraft I've already talked about. Removing a recombination is simply plugging out a recombination away from the graph and routing the ancestral material as if the recombination had never been there. Adding a recombination is the opposite of that. You take a branch that currently does not end in a recombination, and put a recombination event in there and reattach the kind of, you've now got one extra lineage that you need to reattach somewhere into the graph. Breakpoint adjustment just changes where a breakpoint is within a recombination. This is a slightly bigger version of subtree prune regraft, where instead of regrafting the same lineage back somewhere else in the tree, you resimulate the whole thing from the ancestral recombination graph model. So in principle, you can add more recombination events. And this is just wiggling the times of all of the nodes in the tree. Okay, so some benchmarking of how well or badly the kind of resulting algorithm does out of this. We benchmarked against something called ARGWeaver, which many people in the audience may or may not have heard of. It is a similar kind of Metropolis-Hastings algorithm, but using a simplified model with a lot of kind of discretization and somewhat ad hoc disallowing of certain kinds of recombination events in order to reduce the size of the search space. So it's a much more scalable method, but using a more ugly model, if you like. In particular, the ancestral recombination, the ARGWeaver model cuts long-range correlation across the genome, so it really only looks at correlations in adjacent sites rather than long-range ones. It disallows what's called trapped non-ancestral material. Maybe is it visible to Zoom if I doodle a diagram on the board? Or are they not going to be able to see that? Well, maybe I'll just try to describe it, and well, okay. So trapped non-ancestral material essentially means you have a lineage which has ancestral material in its left edge, then a segment of material which is not ancestral, and then more ancestral material afterwards. This thing cannot happen under the ARGWeaver, under the model used by ARGWeaver. That's essentially how it cuts this long-range correlation. And moreover, there's a time discretization involved, which is a bit unpleasant, especially if you have ancient data. So how important is this missing out trapped non-ancestral material? This picture is describing just how much of that actually happens in realizations of ancestral recombination graphs to try to quantify how big the error you make is if you ignore it. The green line is the number of ancestral recombinations. The yellow line is the total number of recombinations, and the red line is the number of non-ancestral recombinations. So this is recombinations happening in trapped non-ancestral material. The picture is on a log scale, which explains why these things don't seem to add up horizontally visually. But the point that's being made, this is a sort of modest genome length scenario. This is where the genome length is very high. And the point is that in both cases, when you increase the number of sites at which you're looking, very quickly, non-ancestral material starts to dominate. So there is a lot of it in typical ancestral recombination graph scenarios. And by ignoring it, you will be disallowing a large number of recombinations, which in principle ought to be there. It's, of course, a further question whether your particular quantity of interest in the analysis you're doing is sensitive to this error. It may not be, but the error is still there. And in principle, it's something that needs to be worried about. It also manifests itself that the same sort of discretization and non-ancestral trapped material errors manifest as biases in parameter estimation. So here we simulated a few scenarios with varying ratios of mutation and recombination rates. So that's what this capital R is, the ratio of mutation rate to recombination rate. And when you're in the setting where you have where you have both of them being about the same, then you can actually see that there starts being a visible discrepancy between parameter estimates you get out of ARGWeaver and parameter estimates you get out of an unbiased method. So the truth is no longer within the interquartile range in the ARGWeaver setting, and it's also underestimating the uncertainty to some extent. As you increase this capital R to 2 or 4, you get into a more and more data-rich scenario. So you have more mutations being informative and relatively fewer recombinations. And the kind of relative performance of the two stabilizes. So if you have a lot of mutations relative to recombination, then probably this bias isn't something you need to worry about. But this capital R equals to 1 is about the sort of human-like regime. And over there, well, whether this is a bias you have to worry about or not, again, I'm sure is an application-specific question, but in principle, there is a detectable bias there. And so we think that's a selling point for having exact ancestral recombination graph-based methods, even if they don't scale particularly as well as ARGWeaver and similar approximations. This slide is studying the impact of the time discretization error in ARGWeaver. So ARGinfer on the left-hand column is our method, and it's inferring a ancestral population size backward in time. So the left-hand end of each diagram is the present day, and this is the dashed line is the true ancestral population size from which we've generated data. And we've run the same analysis on both ARGinfer, our method, and ARGWeaver, the competitor. Both of them do arguably reasonable jobs. The thing that I want to focus on especially is the kind of confidence intervals, these pale red intervals around the posterior mean estimate, and how jagged they are in ARGWeaver. And this is exactly an artifact of the time discretization. These things can only change at discrete time points, and they can have very large jumps there. You get something much smoother from a continuous time model, which doesn't require this discretization. And it's not completely clear visually, but you do get better coverage from this as well. I guess on the bottom diagram, that's more evident. ARGWeaver actually misses a large segment of what's a fairly simple population size profile here because of this discretization artifact. This is output from our ARGinfer. So these are kind of plots you can use to diagnose when a Metropolis-Hastings algorithm is doing well. In an ideal world, they would be just homogeneous blobs. You would see no autocorrelation structure in these blobs at all. That's not quite the case here. So had we run these for 10 times longer, it would have probably come out that way. But it's supposed to give you a sense of the scaling of this algorithm. This is looking at 100 kilobases, 10 samples. So it's a pretty small sample size. On the other hand, it ran in 25 minutes on the laptop on which I'm presenting this now, which is not especially new. I think it's in its 6th year of life, and it was mid-range to begin with. So this is a reasonably scalable computation to this sort of extent. On the other hand, this slide gives you the kind of uglier side of the picture. So ARGinfer, this new method with optimized data structures, what sorts of things will it scale to? You can probably handle 15 sequences of 500 KB on sort of reasonable machines. Any more than that, you'll certainly require big servers or something like that. And the scaling is not especially nice. So the more you go above this kind of, you don't have to go too far above these sorts of sample sizes and problem complexity before you need a very big computer indeed, and then it quickly becomes infeasible. This is much better than earlier Metropolis-Hastings methods, which weren't using these kinds of optimized data structures. So competitor methods, which tend to be somewhat old and run on older machines, but even then there's a sort of very clear difference in the data volume that can be handled. The flip side is that if you look at ARGWeaver, okay, it has these biases, but the data that it can handle is on a completely different scale again. So if you really have a data set that's of this size, then ARGinfer isn't going to help you. I'm down to my last few slides, and these last few slides are about the extent to which DNA sequence data, at least from human-like organisms, really doesn't pin down a unique tree. So I think the message is that you really need to worry about the uncertainty in ancestral trees, and that if you just fit one ancestral tree for an organism like this, that's going to bias your results, probably considerably. So these are 4 different methods that are used to fit trees to DNA sequence data. There's QUAG by Anna Ignatieva and co-authors. This is ARGWeaver again. This is TS-Infer, which is Jerome Kelleher, Yan Wong and et al. And here's Relate from Leo Spiedel and co-authors. And this has been fit to a very small data set, just 2.7 KB of Drosophila. And the point is, all of the trees are completely different. Quark and ARGWeaver produce ensembles of trees, so this is just a particular realization from those two. These two methods are deterministic and will always produce the same tree, but the number of different trees, the size of the graph, the timing of the nodes, they're all dramatically different. And this is quite typical. This is not by any means a cherry-picked example. Even if you just simulate synthetic data from an ancestral recombination graph and plug it into these methods, you will see similar discrepancy. Of course, none of these things there's any reason to believe is anything like the true tree. And so if you want robust analyses using tree-based methods where you sort of build this tree sequence and store your data on that, then you really should be looking at using ensembles of trees and averaging over them at a minimum in order to get a sense of the uncertainty in the tree and incorporating that into your analysis. And that's where these kind of sampling-based methods, whether they be ARGinfer, our method, or Quark, or ARGWeaver, or anything which gives you ensembles, have big advantages. On the other hand, again, these TS-infer and relate are massively more scalable than the ensemble-based methods. So it may not always be easy to get ensembles of trees. This is my last slide, and what it is an advert for, well, TS-kit dev, really. This is a title as well as a URL. It's a consortium of software by now, which has methods built on top of it, which are using the sorts of data structures that I've been talking about. So I've spoken about ARGinfer in particular, which is not up here, but these things are more scalable algorithms built on top of essentially the same data structure, and scalable to really whole genome data from large samples. So TS-kit is the underlying data structure, which also has a large library of analysis functions, which can take one of these tree sequences as input, and you can perform analysis fairly easily using a well-documented library of Python functions. If your data is stored in the tree sequence format. MS Prime is a simulator which simulates ancestral graphs for from the ancestral recombination graph, as well as a bunch of other models by now, and can generate synthetic data from them. TS-infer can take SNP data and give you a fitted ancestral graph, a fitted tree sequence to that data set. TS-date is used for dating the nodes in TS-infer. So TS-infer output by default doesn't come with node times, but TS-date is a further wrapper that can fix that for you. Slim samples from a bunch of finite population models and comes from a slightly different place, Ben Holler's lab. But there is nowadays an interface called PySlim, which interpolates between Slim and the rest of the tree sequence-based methods. And finally, forward Pi11 is a similar sort of forward in time finite population model simulator, similar to Slim, which also interacts with the tree sequence output. So the closing message, I suppose, is actually doing principal inference with recombinant genealogies scales incredibly poorly. You can do clever things with data structures to an extent to improve your scalability, but there is a fundamental limit beyond which it won't go. On the other hand, more scalable methods tend to have the downside that they can't quantify the uncertainty in genealogies, which we have good reason to think is considerable, but that hopefully the kind of library of tools that's beginning to be out there will continue to address that problem going forward. And I think these are the sorts of tools that have a chance of doing that. Thank you very much for your attention.